everybody, and welcome to this presentation from Labskin. Today, we're going to talk a little bit about our new venture, which is Skin Trust Club. Before we talk about what Skin Trust Club is, I guess we should talk a little bit about Labskin and the origins of Labskin and how we came to discover Skin Trust Club. Today, we'll be joined by some of my colleagues. So there's James O'Sullivan, who is the Chief Technology Officer for Skin Trust Club and indeed for Labskin. We have Colin O'Sullivan, who's the Chief Information Officer. And also, last but definitely not least, we have our Head of R&D, David Caballero, who's going to talk to you a little bit about the science behind Skin Trust Club. So first of all, Labskin, as many of you probably know, and hopefully you should know by now, we produce a full thickness human skin equivalent. Our model is basically designed with the skin microbiome in mind. We're very much lovers of the skin microbiome and aficionados of the skin microbiome and all of the good stuff that it does. In terms of where we came to Skin Trust Club. So a lot of our clients would have tested cosmetics with us in the past. So we would have a deep understanding of skin and indeed the technology behind skin science. So it seemed like a natural evolution for us to go and to look at the microbiome on a deeper level and indeed on a more widespread level. So obviously we're looking at the consumer and how the consumer is the consumer microbiome, and then obviously the data points that are associated with the consumer microbiome, such as the environment. So without further ado, I'm going to hand you over to our Chief Technology Officer, James O'Sullivan, and he's going to talk to you a little bit about what Skin Trust Club is and also the application and the science, a little bit about the technology behind it. So James, if you want to go ahead. You're on mute, James.
Sure. Sorry, Eric. I guess it would make sense to speak a little bit just about Skin Trust and the premise behind the idea. So really what Skin Trust Club is essentially a home test kit. We will send you a microbiome test kit where you can swab the cheek of your face and send back the data. And while you do that, actually, our mobile app will capture the environment that you're basically living in. So how it does that actually is it just uses the air quality index on your phone that you're carrying around with you all the time, and it takes various data points from that, such as sulfur dioxide, carbon monoxide, and a few other bits and pieces like UV, pollen count, stuff like that. So we're basically capturing your environment and we're coupling that with a microbiome skin swab. We're taking that back to the lab and basically generating a microbiome index to give you some kind of marker on the health of your skin. So if we move to the next slide, Eric, we can basically see how all this works or the mechanics behind it. So essentially you download the app, we'll post you out a skin swab, you take the sample and send it back to us. We calculate your unique skin microbiome score based on the results of that swab and the environment that I just mentioned in terms of how we capture that. And basically, we design a couple of suggestions around skincare routines, and we base those basically on three categories: dry, balanced, and oily skin. And in terms of the actual app, this just gives you just a little bit of a taster, but essentially when you onboard yourself onto our mobile application, we basically capture your location, and in doing so, we capture your air quality index score, which is made up of some constituents, such as I mentioned, like carbon dioxide, sulfur dioxide, and a few other bits and pieces. And basically, we take that metadata and we make sure that we append that to the skin swab that you physically take on your cheek. If we move to the next slide, there's a couple other things we capture on the onboarding, such as the skin conditions, and a few other questions that make sense in terms of just making sure we have some various bits and pieces that we can add to that microbiome score itself. So the next screen will just give you an idea of the physical box itself. So the logistics behind it, we actually send you out this box. You can activate your kit by using our mobile app, and obviously go through the onboarding process that I just spoke about and actually take the physical swab yourself. And if we move to the next slide, we get an idea of the microbiome reports themselves. So as I said, this is kind of an indicator or some kind of marker for your skin health. And essentially it's broken into a couple of parts: summary, which kind of breaks down what we're actually testing, key findings, and some of the microbes. We kind of highlight some of the microbes that will be standing out for you, the environment that was captured in. So as I mentioned, a couple of things about where you're living and in terms of the air quality index scores. And then we'll provide you with some suggestions in terms of, a skincare routine for the morning and for the evening that might make sense. Or you could append that to your existing skincare routine as well. So you can actually see, if you dive into one of those pieces on this particular screen, you can see that, we pull back the key findings here, some of the microbes, and David will speak a little bit about that in a moment in terms of the 16S data and the quality of that data. And then obviously a key part of the report itself as well is the product suggestions. So we essentially suggest some of the products that we've been testing over many, many years in terms of a morning routine and an evening routine. And these are just suggestions really that might shift your microbiome state to a more balanced approach, essentially. But all in all, I mean, the real purpose behind Skin Trust Club really is that we're trying to solve just a couple of problems. One being that whenever we went in search of data sets in this space to do a little bit of work around the microbiome or something like that, or even skin conditions and things, it was just limited. So we were limited always to small data sets. So trying to actually create a robust data set of the microbiome is really one of the key focuses of this, so that in the future we can do lots of research into this space and actually have large data sets to work some AI into, etc. And then the other one really was, the other big problem for people was generally in this space, a lot of it is observations. So people look at, in the mirror or look at themselves and make a judgment on their skin type. And, when you look at other areas such as fitness and so on, we've all these cool things that we can track data on, we give markers on, and how in terms of how we're getting fit and so on. But when it comes to skincare, the science kind of matters, but there is no real science out there in terms of testing your skincare, routines, and if they're really working or not. And I'll pass this over now in terms of how that metadata is handled and processed to Colin. Thanks.
Thanks, James. So, as James pointed out, a big challenge with the microbiome is getting those, I suppose, high quality data sets, and you really do need those high quality data sets to produce good models. And looking at the next slide there, you will see there's really three, I suppose, broad categories that the data will fall into. The first being the microbiome data, which is the 16S sequenced data. Then the second set is the metadata. So, all those details around the person's health, previous skin conditions, their age, their gender, lifestyle data, such as are they a smoker, do they get exercise, all that type of data. And the third set, which we collect, is the environment they're living in. So the humidity, the average UV index for that area that they're living in. Is it urban? Is it rural? Their product usage. And, the combination of those, I suppose, three categories, when they intersect, you have a very powerful, analysis data set that you can produce really accurate models with. And moving on to the next slide, this shows you some nodes, and basically each one of these nodes is where some action takes place on the data. And if you look on the left-hand side, you'll see the Skin Trust Club app has been installed by the user. And of course, they're using that on a daily basis. It's installed on their device natively. And of course, we can gather the local data, so the pollution scores for that day, the average UV index scores, humidity, all of that, I suppose, good data that we get in real time. And as you can see, the blue line across the bottom there, that the environment data is being fed to us in real time as the user uses the app. And if you go across the middle, you'll see the test activation node is a key node where the customer receives their kit, opens their kit, and scans their QR code, and that's an activation point. It's a chance for us to basically timestamp the data. And so it's a chance to, I suppose, align the data sets between the environment data that's happening at that time, the actual test kit, and of course, the metadata that they've provided to us as part of that confirmation on the activation. That all those data sets are obviously controlled by us through our app and our infrastructure, and essentially we're able to validate the data sets against very exact data schemas. And so it allows us to curate the data in a way that wouldn't really be possible if we didn't have this system from end to end. And you see one of the big nodes there is the data cleansing node in the middle, where it all comes together. And essentially that data cleansing node allows us to process these data sets and basically store that into a research data lake, where from that repository, we're able to build advanced models to analyze the microbiome data and take into point all the other data points we have as well. And I suppose you can see there's a natural iterative loop there for evolving our models. So as our data set increases in size, we obviously will send out new versions of our models to the app. And so we're going to have this improving analysis model happening all of the time, which is very exciting for us. Of course, the user gets their results in that final node. But something I didn't mention, and you can see that node at the very top, and that's the ongoing research that we do at Labskin is obviously very important to us. And the data sets from this application and being able to control the data in this very, I suppose, unusual way gives us really great opportunities to use the data. And so it's a very exciting opportunity for us. And I'd like to pass off to David to talk through the science.
Okay, thank you, Colin. Well, I will give you some details how and why we do this. The why is the microbiome, which is the whole population of microorganisms that live in our skin, are actually linked to certain states and even conditions or disease of the skin. Most of the microbes are beneficial, that live in our skin are beneficial for us. And they form a protective layer that help us against UV, pollution, and some other insults that come from the environment. But different composition, different ratios between them and the presence or absence of certain species gives you information of the healthy or of disease state of your skin. So that's why we use the microbiome. The microbiome to establish a relationship with the skin. Basically, our skin and the microbiome talk to each other, and they are in an equilibrium that needs to be kept to have a healthy skin. We are established and validated a method to understand the microbiome, but we are currently working on even improving this system to look at other things like metabolizing skin with lipids, fatty acids are in your skin, gene expression from the skins to see not only how the microbiome, but also how the skin responds. In the terms of the microbiome, in the next slide, for those of you that are not familiar with this sort of analysis, what we do is something called metagenomics to analyze the composition of the microbes on your skin. All bacteria contain a gene named the 16S ribosomal gene. So all have this gene, but these genes contain different regions which are variable between species. They are all these B1 to B9 regions here. So we are able to identify a species of bacteria due to the difference they are in these genes. The way we do this, first we amplify a fragment of that gene with the primers forward and reverse, you can see there. Then we do something called barcoding, which allows us to put nearly 100 samples together from 100 people. We add a little barcode for each of these samples. We can then identify each of the microbiome from each of the person. And we do something called next generation sequencing. The next generation sequencing is a high throughput method that allows us to look at hundreds, if not thousands sometimes, of different microbes that are present in those samples. And then we do data analysis using bioinformatics and AI as AI technology and algorithms like Colin was mentioning. So this is a relatively well-established method, or at least in the last 10 to 15 years. There is always been a lot of variability due to different methods of extraction, methods of sequencing, methods of analysis. So what we wanted to try to achieve here is control and reduce variability. And we have done that by using a very specific workflow. And we have tested different workflows in the labs. In the graph in the left, you can see there are 3 different workflows, A, B, and C. C1 and C2 are the same workflow with different bioinformatics pipeline, different analysis. And you can see that we can identify a number of groups, class, species, that is how the bacteria is classified, with increasing accuracy with the different methods. And we have validated our method C, which we have found is the best, by doing several replicates, and we can identify a species with a ratio of 27%, with a standard deviation of 1%, which is really good. So we have a validated method that is reproducible and transferable to different laboratories. And in this next slide, we say this is a control that we add. How do we know that where we are analyzing on your skin is they are real microbes? There is no an artifact of the method, so we always include controls. We mix several microbes that are well known to be on the skin in a control that then we can check if we can identify them using our technique. And in the method C here, you can see that from the 6 mix on top to the method C in the bottom right, we identify all of them to the genus level and 4 of them to the species level, which is actually very good for this sort of metagenomic analysis. And just to show you the last slide, this is initial report that you will see when you are in the app with the ratios, the composition of your microbiome to different levels. You can go in and see several species, but also the genus. And you will have a classification with a microbiome index, which is the balance of your microbiome that depends on certain aspects of the microbiome, biodiversity, the presence of certain groups, the absence of certain groups. We mix several things together in a fairly complex algorithms that give you that balance score. And we can, using those scores and some other features from the microbiome, we can give you certain characteristics of your skin, like if it's a dry or normal or a sebaceous. And also we can go into the detail in this little heat map here. You can see how different type of skins that has been classified using this method gives you different species. The amount of that is the presence of accent or how much of that species is in your skin. For instance, what is well known, Cutibacterium acnes is a very high in numbers in sebaceous regions and also in sebaceous skin because it feeds in the sebum of the skin. It's related to acne vulgaris. Then you have things like Staphylococcus epidermidis, which is a well-known good guy on the skin, and this is one of the main components of our microbiome, but also smaller things like Lactobacillus inner, which is a probiotic, a well-known probiotic of the guts, but it has also been recently shown to be present on the skin and is known to be absent in people that have psoriasis and eczema. So we can correlate certain things with the fetus on your skin. And I think I will leave it here for questions on later.
Yeah, so I'll just do a quick summary, I guess, because I think there's a bit of everything in that presentation for something for everyone. So yeah, as I mentioned at the beginning, we obviously we're Labskin, and this is now Skin Trust Club, which we're presenting. We came from producing a human skin equivalent and doing cosmetic tests on human skin model for over the years, to identifying a number of problems which we wanted to solve, which, obviously Skin Trust Club, it gives the consumer information about their skin, gives them knowledge about how their skin is performing. It also gives cosmetic companies the opportunity to test their products against our Skin Trust Club, and to have their products recommended based on data and based on science. And through this whole process, we'll be gathering, very useful data around the skin microbiome, and also the overlapping metadata points such as environment, such as age, and all of this kind of stuff to build up what we would say as a first of its kind database in the microbiome space. So it's a very, it's a very intriguing project. We're very excited about it, and we're talking to partners at the moment, having conversations about how they can participate in Skin Trust Club in various ways. So as I said, if you have any questions or you want to follow up the presentation, feel free to reach out, and we'll be more than happy to help. And thanks to the lads for a very engaging and a very informative presentation.